Welcome everybody to the January 2023 edition of klogW. This is part of a mini series of talks celebrating the 2022 APS-GSNP Fellows. So we had Santo Fortunato speak in December, and that recording actually will soon be available. I say it's already available, but I haven't put it up yet. Today's speaker, of course, is Jacinta Conrad, and I'll introduce her in a second. And on February 24th of this year, we will have Shrikant Shastri from Jawaharlal Nehru Center for, what is it? Advanced Scientific Research in Bengaluru. Just a few things before we get to the talk. I would like to take this opportunity to encourage everybody, if you're not already a member of GSNP, to please become a member. This gives you access to prizes and awards for both students and faculty, and participation in invited and focus sessions at the upcoming March meeting. And also, it gives you a chance to attend and contribute to events like this series, as well as other networking and professional events. So to become a member of GSNP, if you go to the website, APS website, you can just search APS-GSNP and you will get to this Engage website. And there's a button there that says Join GSNP. And the last thing I want to say is just a little bit of etiquette for this talk. Our etiquette is like many other talks that you've been to over these past 3 years, that during the talk, please make sure you're on mute. During the talk, if there are clarificatory questions, please post them in the chat, and Daphne, when she joins me, will ask JC those questions. So JC, you don't have to monitor the chat box. We'll ask questions. Otherwise, if they are broader and more involved questions, we might save them till the end. At the end of the talk, if you put your virtual hand up, I will call on you in the order that you raise your hand, and you can unmute yourself and ask your question yourself. Or if you prefer that I ask it, you can of course still post it in the chat box, and then I will read the question out for you. Okay, I'm going to stop sharing this now. And let me switch to introducing JC. So JC Conrad is a professor at the University of Houston. She's the Frank M. Tiller Professor of Chemical and Biomolecular Engineering there. She is also the chair of the American Physical Society's Division for Soft Matter Physics. She is a fellow of both the American Physical Society as well as the Society of Rheology, as well as an executive editor for ACS Applied Nanomaterials and a member of the Soft Matter Advisory Board. JC got her PhD from Harvard University. Well, let's say you got your bachelor's in mathematics from University of Chicago and your PhD from Harvard University, and worked as a postdoc at UIUC, University of Illinois at Urbana-Champaign, before joining her faculty position. I want to end by reading her APS fellowship citation, which reads, "For experimental contributions to understanding nanoparticle dynamics, bacterial adhesion, and colloid polymer mixtures using advanced microscopy and light scattering techniques." And JC is going to tell us today about nanoparticle transport in complex fluids. So I'm really looking forward to this. JC, the floor is yours.
Okay, sorry. Give me one moment because I apparently failed. Okay. So first of all, thanks to everybody who's attending and everybody who watches this later. I want to echo something Shubha said, and I say this as a unit leader right now in APS. Please do join APS units. Please join GSNP. It's actually very important for supporting programming and supporting awards and travel. So again, please do join units, join all of them, I think, but especially join GSNP. Okay, so I'm going to talk today about nanoparticle transport in complex media, and specifically talking a lot about diffusion. And I want to highlight, first of all, the students who've carried out this work over years, Firuze Babayakharasani, Brian Poling-Spetvik, Max Smith, Ali Slim, and Farshad Safi Samgabari, as well as a number of collaborators over the time that we've been doing this work. So let's see, first, I'm going to talk about diffusion. So we generally know that diffusion starts with Brown, who observed that the motion of small particles in a liquid was jiggly and irregular. Okay. And so I'm showing here just, again, a sort of a throwback movie to looking at pollen grains inside a liquid medium. And from a statistical standpoint, this process can be described, for example, by measuring a mean square displacement, which I show the calculation here. And if I look now at the mean square displacement for the small particles in the previous movie, we expect that for normal diffusion, that the mean square displacement should scale linearly as a function of the lag time delta t. And the slope of that line then gives us the diffusivity, which characterizes the extent of the motion. So that proportionality constant gives you the diffusivity. And a final comment is that for normal diffusion, the distribution of particle displacements is Gaussian. Okay. I'm going to talk today about complex mixtures. And these are of interest because they appear in a wide variety of practical settings and applications. For example, nanoparticles delivered through the body are important for drug delivery and targeted delivery to specific tissues. They appear as novel adhesives. They're extremely important for the processing of advanced materials and nanocomposites. And there's an increasing interest in understanding how complex mixtures flow in porous media, both for practical applications and composite processing, as well as for other applications that involve subground transport. Okay. And so the common theme of many of these settings is that we're interested in trying to understand and predict how nanoparticles are able to diffuse and spread. Okay. So typically, when thinking about transport of particles in complex media, the first question that folks ask is, are we measuring a property that reflects the bulk viscoelastic properties of a medium? Okay. So it's possible now to take this Stokes-Einstein relationship that was developed for simple fluids and generalize that to be able to relate now the mean square displacement of a particle to the complex viscosity of a medium. Okay. And this is called the framework of microrheology, originally developed by Tom Mason. And I show that here. So I know that going from the thermal probe motion to the linear viscoelasticity of the medium has two really critical assumptions here that we'll talk about. First, making this assumption requires that one assumes that the material is a homogeneous continuum, as well as it's isotropic and compressible. But it's really this idea that we have a material that's both a continuum and homogeneous that allows us to make this assumption. So now I want to think now about what happens when we put very small particles or nanoparticles inside a complex medium, or here this is a polymer melt. Okay. So original work from the groups of Michael Mackey and Michael Wong showed that if you measure the diffusivity now of nanoparticles in a polymer melt, okay, that the diffusivity is much faster than one would expect based on the Stokes-Einstein equation. And so you can see that this is up to a factor of about 100 times faster than you expect based on the viscosity. Okay, where here the comparison is to the true viscosity. Okay, what we would measure in a rheometer. So why is this interesting? So the first thing that you can see is that nanoparticle diffusion doesn't reflect the continuum properties of complex solutions. Okay? Indeed, there's a mystery. Particles often diffuse much faster than expected. And the second implication then is that you can imagine now that the viscosity that's felt by nanoparticles is much lower than you would expect from a bulk mixture. So how does one start to understand the properties of these systems? Well, this is an experimental challenge in that many methods often access average behavior and neglect any concepts of heterogeneity. And there's a separation in time and also length scales between the solvent, solution, and particles in many complex mixtures. And a final challenge is that, of course, the medium itself may have interesting and time-dependent structure and dynamics. So we've started to think about this problem by trying to come up with systems that allow us to both understand the structure and dynamics of the medium, and then relate that back to the dynamics of the particle. And the systems that we work with are polymer solutions. We like polymer solutions because we can tune both length and time scales associated with structure and dynamics by controlling the size or molecular weight of the polymers as well as their concentration. So first, from a structural standpoint, in a dilute mixture, the polymer coil, a flexible polymer, adopts a Gaussian conformation that can be characterized by one length scale, the radius of gyration Rg. Now, as we increase the concentration of the polymers, we find that eventually we increase it so much that the polymer coils begin to touch. This happens at what's called the overlap concentration. And then above the overlap concentration appears a second length scale, which is the average separation between the polymer chains, which is called the correlation length, or C. I also note that the dynamics now exhibit similar transitions. If we measure the dynamics of polymers, you find that the mean square displacement also scales now as a power law with an exponent alpha. And the value of that exponent depends on the physics that goes into each particular regime. Okay, so in the dilute regime, the exponent is 2/3. In the semi-dilute regime, where hydrodynamic interactions become screened over the correlation length, the exponent is 1/2. And then finally, in an entangled regime, now polymers are free to slide along tubes that are formed by their neighbors, and that exponent becomes 1/4. Okay. So using then the predictions of polymer physics, we can understand the structure and dynamics of these of polymer solutions. And now we can use this as a test bed to try to understand how particle dynamics couple to these features. Okay. So of course, I should note one other thing. We were interested particularly in size and length scales, where this continuum assumption that underpins the generalized Stokes-Einstein relationship begins to break down. Okay. So I've told you already about the microrheology framework, where again, here it's assumed that the particle size is much greater than the length and time scales that appear in that medium. And so here particle dynamics probe the bulk viscoelasticity of the solution. If we make the particles very small compared to characteristic length and time scales in the solution, now we approximately probe the solvent viscosity. But of course, the regime that we're interested in is when everything is happening on similar length and time scales. When particles are of similar size to characteristic length scales in that solution, now we can no longer treat the medium as a homogeneous continuum. And that's where we'll be playing. Okay. So we started out doing very simple experiments. We put particles of different sizes inside well-controlled polymer solutions. Okay. Here, the polymer solutions that we originally studied are partially hydrolyzed polyacrylamide. Okay. This is an interesting polymer, mostly because you can get it at very high molecular weights. It's used typically for enhanced oil recovery, but we're able to get it in molecular weights that are comparable then, that give us comparable radius of gyration in dilute solution to the sizes of particles that we can image and track with standard microscopy methods. So here I show diffusion of particles. One of the movies that we take, you can see that particles are again jiggling around. And from those kinds of movies, we can follow the motions of particles over time and calculate a mean square displacement as a function of lag time. Okay. So here I show a representative set of data. This is where we fix the particle size here at 600 nanometers. And as we go from left to right, we increase the concentration of the polymer solution, going from below the overlap concentration to above the overlap concentration. What I want you to notice is that, first of all, the dynamics slow down as we increase concentration. That makes a lot of sense. We know that we expect that because everything gets more crowded. But these lines have two regimes of behavior. Okay. On long time scales, we find that the scaling exponent is 1. That allows us to define a diffusivity that we can extract out from the slope of that line. But on short time scales, we find that the mean square displacement scales as a power law with an exponent that's not 1. And so we characterize that through the value of that exponent. And I'll talk about both. One thing I want to note, though, is that this is really not probing the dynamics of the medium. Okay, we can go back and use that microrheology framework with different size particles. And even our 2 micron particles, if we grind the microrheology analytical platform, okay, and we calculate from the MSD the viscous and elastic moduli, okay, what we find is that we always see a deviation between what we measure in microrheology and what we obtain from bulk rheology experiments. Okay, so we're not doing microrheology. We are instead really accessing something about the dynamics and structure of these polymer solutions. Okay.
JC, can I ask you quickly about that previous 2 graphs ago? This one, yeah. So you're going to 26 C star. So that's, you're changing the concentration.
Yeah.
Is this the concentration of polymers to 26 times the critical?
Yeah, this is, so we go from below, I tried to say that, but I'll say it more slowly. We go from below the overlap concentration. So C star here is when the polymer coils begin to touch to somewhat above it. And then some technical details here, the solutions are slightly charged. And so it turns out that the entanglement, these solutions are overlapping, but not entangled.
So this is not straight off diffusive behavior, and they're all curved, right, on this log-log plot?
Right, they're all curved. Well, we have two regimes. We get a well-defined diffusive regime on long time scales, and we get a different regime on short time scales. And then there's a crossover that we spent a long time trying to analyze and never got anything very satisfactory for. But we do seem to see well-defined slopes at short times and long times.
And what is C star typically?
Oh gosh, I should know that number and I don't. I think for these, it's on the order of milligrams per milliliter, but don't quote me on that.
Okay. There's also a question in the chat. What is meant by hydrolyzed polyacrylamide? There are no hydrolyzable bonds in polyacrylamide.
Hold on, let me go back to it. So this is the structure of what we look at. And so I think that the partially hydrolyzed refers to the difference between the CONH2 groups and the COO groups where we now can attach. Yeah, sorry, I'm not a good chemist, but that's the chemical structure of what we're looking at. Does that answer the question? And I think what this also means is that whether or not these groups are fully ionized or not varies for these systems. It turns out for various other reasons, this is a terrible polymer to use, but because of the fact that you have a fraction of the charge groups that are ionized along the backbone. And I can talk a little bit, well, I don't have the slides with me here to discuss this, but I can talk a little bit about what the implications of that mean. Basically, the viscosity of these systems doesn't scale cleanly as expected for either a charged polymer solution or a neutral polymer solution. It's something in between. Does that answer the question?
So do you mean the proportion of sodium carboxylate versus amide substituted groups?
Yes, and that's fair.
Thank you.
Thank you. That's a much cleaner way. I'm sorry, I'm a terrible chemist. So thank you. That's a much cleaner way of saying what I was trying to say. Great, thanks. Okay. Okay, so great. So what we do is we can first look at the value of the diffusivity that we extract out on long time scales. And so what I plot on the left is the relative diffusivity, where we normalize by the diffusivity of particles in the solvent. Okay. And we do that for different sized particles. I'm going to actually go back quickly just to focus. Okay. And so we do this for different sized particles at different polymer concentrations. Okay. And what I'm plotting as open symbols is what we would predict for the diffusivity of these systems if we were measuring, if we took the bulk viscosity and used Stokes-Einstein. Okay. So what I want you to notice from this plot is that the relative diffusivity is always greater than the Stokes-Einstein prediction, and that can be up to about a factor of around 30 greater. Okay. You can see this a little bit more cleanly if I instead plot a normalized diffusivity, as I'm now normalizing by the Stokes-Einstein diffusivity rather than by the diffusivity of particles in solvent. Okay. And what you see from that is that as we increase either the polymer concentration or we decrease the particle size, we see increasing deviations from that Stokes-Einstein prediction. Okay, so we can see that now as we make the particles smaller, so we look less and less continuum, we start to see even greater deviations from Stokes-Einstein. Okay. So we've tested a number of models and pictures that would allow us to understand these dynamics, but I'm only going to talk about the one that appears to best describe our data in these conditions. Okay. And so this is a coupling model, it's due to Michael Rubinstein, Li-Heng Tsai, and collaborators. Okay. And the physics of this model is that the particle dynamics are coupled to relaxations of polymers over a similar size. Okay. And so the similar size means that once the polymer relaxes, okay, a segment of the polymer relaxes on the same size scale as the particle, then the particle is able to basically escape a local cage and is free to move. Okay. So the critical predictions of this model then, comparing back to our experimental data, is that first of all, we expect that the particle motion is coupled to the Rouse dynamics of the polymer. So that would give us an exponent of 0.5 on short time scales. And then second, the long time diffusivity can be collapsed as a function of a scaling parameter, which is the radius of the nanoparticles over the correlation length with an exponent of -2. Okay, so those are the predictions we're going to test. First, we test the diffusivity prediction. So this is again looking at a variety of different particle sizes and polymer concentrations. That's what leads to changes in the correlation length. And we find that over the range of data that we were able to access, that we get reasonable agreement with that scaling prediction, which is shown here as a black line. Note that as we make the particles bigger, we start to fall off that line. That's because we start to move into a more continuum-ish limit. Okay. So the first conclusion that we can make is that coupling to polymer dynamics on the correlation length is what appears to control the long-time diffusivity of the particles in the system. But if we go in and we look at the value of that subdiffusive exponent, okay, the model predicts 0.5, which I'm showing here as a dashed line. Okay. Our large particles appear to approximately follow that model, but our small particles deviate from it. Okay. So this was puzzling and interesting. And of course, so now we start to run across some of those experimental challenges. In the very simple particle tracking experiments that I've shown you so far, we're able to look at the particle motion, but not the polymer motion. So we started to collaborate with my colleague, Jeremy Palmer, who's a computational scientist, and we developed different types of molecular dynamics simulations that would allow us to access now the dynamics of the polymers as well as those of the particles. And the most interesting thing that we get out of those simulations is that we can look for coupling between the dynamics of the nanoparticles and coupling now between the polymer center of mass. So what I'm plotting here is the subdiffusive exponent of the nanoparticle. That's what we extract from the mean square displacement, now as a function of the subdiffusive exponent of the polymer center of mass. What I want you to notice is that all of these points fall on a line. The different color families here refer to two different types of simulations, one with explicit long-range hydrodynamic interactions and one without long-range hydrodynamic interactions, which don't seem to strongly matter here. But we always find that that subdiffusive exponent reflects the subdiffusive exponent of the polymer center of mass. Okay. And so what does that tell us? It tells us that in addition to coupling to the segmental relaxations in these systems, there's also a coupling to the center of mass motion. And we can understand that as thinking of this sort of like a traffic jam or like, I live in Houston, so we think about traffic jams a lot, right? So I need to be, if I want to try to escape in a traffic jam, I need the car in front of me to be able to move. And that's how approximately we think about the center of mass motion. That gives when the center of mass of the polymer moves, and it moves on similar timescales to the particle because they're of similar size, okay, that gives another mechanism that allows the particle to escape its local environment. And so what we probe in our experiments and simulations then appears to be this combined coupling mode, both to the segmental relaxations as well as to the center of mass motion. Okay, so I gave you a physical picture, which is really nice. And now I'm going to talk about all the ways that we've tried to break that. Okay. And so I gave, this is this, what I've told you about so far is basically the spherical cow of our experiments. We picked, well, a relatively simple polymer, and we picked a relatively simple particle. And I'm going to go through now and talk about different ways that we can think about asking whether or not this type of physical picture is relevant for a number of different scenarios. And so I'm going to talk about 5, which are shown here on the right. Okay. I'm going to talk about changing how the particles interact, either by shape or by how they interact with the medium. I'm going to talk about how the polymers might change, either through their morphology or through their flexibility. Okay. I'm going to start with looking at particle-particle interactions. So in our first experiments, we had dilute particles, and we assumed that they had roughly hard sphere interactions. Okay. But experimentally, we were also interested in trying to overcome the challenge of thinking about how to access both particle and polymer dynamics. And so we developed another model system that would be compatible with different scattering methods. Okay. So the system that we developed is now silicon nanoparticles that were in a mixture of, where we added polystyrene polymers, which are non-absorbing, in a solvent to butanone that we could either fully protonate or partially deuterate. And this is important because the system is interesting in that we can isolate the particles through one scattering method using X-rays, and we can isolate the polymer using another scattering method, which is neutrons, okay? And by changing the protonation or deuteration of the solvent. And so now, if we do two different sets of scattering experiments, we can look both at particle structure and particle dynamics, and polymer structure and polymer dynamics. And so for the non-scatterers in the audience, we make just the types of measurements we can make. We can make static measurements where we measure the intensity as a function of a wave vector Q. We also make dynamic measurements where we measure basically an intensity autocorrelation function, now as a function of a lag time and also of the wave vector Q. And we note that the scattering vector Q probes the length scale 2 pi over Q. Okay. Great. So I'm going to again just sort of talk about highlights. Here I'm going to talk about the particle dynamics. So we measure those using a technique called X-ray photon correlation spectroscopy. I'm showing those correlation curves here on the left for a particular system. This is 3% silica and 10% polystyrene in our solvent. And what we find is that the curves in this case can be fit to stretched exponentials with an approximately constant exponent of 2/3. And if we compare back to our microscopy experiments for these concentrations and length scales, that's the same exponent. So we're measuring the same physics now on different length scales for these systems. So from these fits, we can extract out a characteristic relaxation rate, which I've indicated with a dashed line here. And then we can do experiments over different concentrations of particles, where now we added more and more particles to look at denser and denser systems. And we can also, again, vary the concentration of the background polymer. Okay, so again, some physics. If we're now in the length scales that we access with scattering, which are different than those in microscopy, okay, and with a stretching exponent of 2, if we're accessing the Zim dynamics of these systems, we expect that that the relaxation rate should scale as a function of Q cubed, okay, which is the line that I've drawn on the plot. What I want you to see, though, is that we're not measuring Q cubed, right? And instead, there's a particular set of length scales where we seem to measure something that's quite different, which I've indicated by the dashed line here. Okay, so how do we understand this physics? Well, again, note that we have the ability to access both structure and dynamics in these systems. Okay, so what I'm plotting here is the static structure factor that we obtained from the static scattering measurements. What we find is that there is a characteristic peak in our static structure factor. This tells us that there's a characteristic separation between the particles. Okay. And now on top of that, I'm going to plot basically 1 over an effective kinetic constant. Okay. It's something like a diffusivity, but it's not. We're in a subdiffusive regime, but we're defining something that looks kind of like a diffusivity. I've called it a subdiffusion coefficient here. Okay. So I'm going to plot that now on top of this data. And what I want you to notice is that 1 over K, this subdiffusion coefficient, really cleanly follows the structure factor in our systems. Okay. So how do we understand this? This is a phenomenon called de Gennes narrowing. Okay. And the physics of de Gennes narrowing is straightforward. Okay. A peak in a structure factor tells us about a local energy minimum in our system. And then de Gennes narrowing tells us that it's slower to diffuse out of that local energy minimum. And this is what gives us the inverse coupling between the static structure factor and then the subdiffusive coefficient. And so it's interesting because typically de Gennes narrowing is seen in very concentrated systems, where here our particles are still relatively dilute. We're working at weight fractions of only 10%, so volume fractions of about 5%. Okay, so what this tells us now is that we can start to see coupling that arises both from the interactions between the particles, here they're electrostatic with the medium, and that arise due to concentration when they start to feel each other. Okay, so a second thing that we can look at is particle polymer interactions. So how do we tune that? Well, I did my PhD on colloids, which we think about hard particles, okay? And I'm showing here, if I were to just make a prototypical plot of the colloidal viscosity as a function of a concentration, okay? We know that the viscosity of hard colloids diverges at some concentration or volume fraction. Okay? Polymers are soft and deformable, okay? And so if I were to make a similar plot about how the viscosity of a polymer solution changes, we would find there's no strong divergence, and instead that increase happens more gradually. So how do we now think about changing the interactions between particles and polymers? Well, one way to do this is to attach polymers to the surface of our particles. Okay? And so now this gives us a squishy or deformable object. These are called polymer-grafted nanoparticles. They have physical interactions with our polymer matrices. And practically, these are of interest because if you measure, for example, the elastic and viscous moduli in these systems, this is a graph from Lyndon Archer's group by Sam Srivastava, who's now a professor at UCLA, you can find that adding in these grafted nanoparticles changes these bulk relaxation, the bulk viscoelasticity of the solution. And so the mechanisms on the microscope scale by which this happens are not well understood. Okay. So we now take the same silica system that we developed for our scattering experiments, and we graft polystyrene chains to the surface. Okay. We graft them at fixed density. We vary now, and we attach mostly 1 molecular weight of polymer on that surface. I've given the parameters here. And now we add our polymer-grafted nanoparticles to solutions of polymers of various molecular weights and at various concentrations. So the first thing I want to measure, we first just measure the bulk viscosity of the solution. Here I'm plotting the specific viscosity as a function of concentration. Okay. What I want you to notice that if we plot as a function of a concentration normalized by overlap, that we should take care of all of the differences in the length scale dependence in our polymer solutions. And so all of our points for the different molecular weights should fall on top of each other, which they do in this top right-hand corner. I also put the inset of the specific viscosity as a function of the shear rate to show that these are really just liquids. Okay, the viscosity is constant, even at our highest concentrations. Finally, we can also look at the scaling exponents, which we can compare back to polymer theory, and we find that we see the scaling exponents that we would expect for the different regimes of behavior. So we have a semi-dilute unentangled regime, okay, where we expect that the specific viscosity scales as a power law of 2 with a normalized concentration, and then the semi-dilute entangled regime. And we can see that crossover from the semi-dilute unentangled to the semi-dilute entangled regime in the bulk measurements. Okay, great. So we were interested, these are the bulk measurements, we're interested always in local dynamics. So we put these solutions now back in scattering. And so by, again, selectively, and we can measure the dynamics of the grafted particles, again, using X-rays. Okay. And so that's what I'm showing here, the same kinds of measurements I just showed you, and we extract out from the intensity autocorrelation function a relaxation rate. And we do this for varying concentrations of the free polymer. And so the first plot that I'm showing you is the relaxation rate. Okay. We fix the concentration of the grafted nanoparticles, and I increase the concentration of the free polymer. We find that the relaxation rate slows down as we increase the polymer concentration. This is what we would expect. What's interesting and surprising, however, is what happens when we change now the molecular weight of the free polymer. Okay. Now I'm making measurements at a constant normalized concentration of free polymer. What we find is that the relaxation rate increases as we increase the molecular weight of the free polymer. And why is that weird? Well, I just showed you specific viscosity measurements that said that when we plotted things as a function of a normalized concentration, there's no difference in the bulk system, but there are differences in the nanoscale dynamics of our grafted nanoparticles. So there's something very different now about how this grafted object is interacting with the polymer matrix as a function of the free polymer molecular weight. Okay, fab. So we can make the same kinds of comparisons that we had done in our previous systems. We plot, for example, the long time diffusivity of the grafted nanoparticles as a function of that free polymer, as a function of the polymer concentration, the free polymer concentration. Okay. We can compare it back to the Stokes-Einstein prediction. That's the open symbols. We find that the dynamics of the grafted nanoparticles are always faster than what we would obtain from Stokes-Einstein. Okay. This is what we also saw for our hard sphere systems. But now if we try to compare back to our hard sphere model, where we expected that we could plot everything as a function of the particle of the diffusing object size relative to the polymer solution correlation length, we no longer see a clean collapse of our data, and we don't see that our data follow that -2 prediction. So the take-home message is that our grafted nanoparticles diffuse faster than we expect in those high molecular weight solutions. Okay, so how do we start to understand that? The short answer is we don't perfectly, but my student was really clever, and he said, well, is it possible to think about controlling those interactions by comparing the size of the grafted polymers on our nanoparticles to the size of the molecular weight of the polymer in solution, or the molecular weight of the polymer in solution? Okay. So on our scaling curve, he introduced a factor that is the ratio of the molecular weight of the free polymer to the molecular weight of the grafted nanoparticles to some exponent. Okay. And I'm going to say that he varied that exponent. And what we found is that if you chose an exponent of -1/8, okay, and it's really -1/8, it is not -2/5, it is not -1/3, okay, that we really cleanly collapse all of our data across different free polymer molecular weights and different concentrations onto a single curve. And then if we have reviewers who ask, well, if you change the molecular weight of the grafted polymer, do you still see the same collapse? For a more limited number of samples, now with a smaller grafted polymer, we find that they follow also the same curve. And now if we take this -1/8 dependence and we propagate through that scaling model from the Rubinstein group, okay, it turns into a 3/8 dependence if you account for the corrections. And we find that we see something where we can, again, approximately collapse all of our data onto a curve as a function of this normalized length scale, now with this correction for the difference between free polymer and grafted polymer molecular weights. Okay, so this is rather technical. I'll just note that the hard sphere predictions here really overcorrect for the polymer grafted nanoparticles. Okay. So how do we understand physically what's happening? I've shown you that we can make these collapses. We have no rationale for the -1/8. We've asked many people. If you ever come up with one, please let me know. We have no rationale other than it does seem to be very cleanly -1/8. Okay. So a couple of things. One I'll note is that we see that -2 behavior sort of in this intermediate regime of the scaling parameter. We start to fall off of it at high values of the scaling parameter. This is likely because we can see the onset of entanglements in our solution. And again, this is somewhat unexpected because entanglements don't affect the dynamics of small hard spheres, but they do affect the dynamics of these grafted objects. Okay. So the physical picture that we have to try to explain these systems is to think about what the surface of this object looks like. Okay. So this would be my hard sphere. This is a cartoon in a polymer solution. Okay. The polymers can get effectively very close to the surface of that object. And then we feel if we were to look at a force versus distance curve, it would be relatively sharp between two particles. Okay. So now what happens when I attach grafted polymers to the surface? Okay. The ability of the free polymers to get close to the surface of our nanoparticle depends on the size of the free polymer relative to the size of the grafted polymer. Okay. Small polymers are able to penetrate that grafted corona, leading to a relatively soft interaction. Okay. Large polymers, however, are not able to penetrate that grafted corona. And instead, okay, the corona of the graft remains relatively free of the polymers in that case. And so what this means is that this helps us understand then that what we're seeing is something that's akin to a change in the amount of viscous drag that's felt by these systems, okay? Where we get a non-draining Zim-like regime at high molecular weight, and we get a more Rouse-like draining regime at low molecular weight. Okay, and so not finally, I'm going to next talk quickly about now what happens when we start to change properties of the polymers. Okay. So I told you we set up some simulations. Well, the beautiful thing is once the simulations were running, we could start to be very creative and ask different questions. Okay. So for example, we were interested in asking whether changing the morphology of our polymers would affect the coupling mechanism that we found in the simulations. And so we looked for a while at simulations of ring polymer systems. And so I'm showing just a snapshot from one of those simulations here. Okay. What we found, though, was rather interesting for us. Okay. If we made comparisons under particular conditions, okay, so we picked systems of ring and linear polymers where their free radius of gyration was very similar. Okay, that meant that we had different numbers of monomers in each case, but their size was similar. And we subsequently defined a correlation length for these systems via a geometric pore size, that we saw no difference in the diffusivity of our particles in ring or linear solutions. So they were insensitive to the difference between these two morphologies, right? The dynamics are simply controlled by these characteristic length scales. Even more interestingly, I'm not going to show you data, but if we concatenated some of those rings, we found very similar results, that the free diffusion did not matter on the presence and number of those ring concatenations. So not all changes in the polymer properties lead to differences in the nanoparticle dynamics. But one change that does matter is to change the stiffness of the polymers. Here I'm showing simulation snapshots. On the left is a more flexible linear polymer. On the right is a less flexible linear polymer. If we look at now the coupling between the nanoparticle subdiffusive exponent and the polymer center of mass subdiffusive exponent that I showed you before, okay? Whereas for our flexible systems, we find that these two were linearly correlated. That's the light up pointing triangles that you see in this plot. As we now change the bending potential in these systems, we find that we start to destroy that coupling. Okay, so the arrow here points in a direction of increasing stiffness. So by changing the flexibility of the polymers, we're able to disrupt this coupling to the center of mass dynamics. Okay, so this is a simulation result. And of course, then we wanted to think about ways we could test this in our experiments. So we didn't have a great answer, but one way we thought about was to now start to change the presence of the charge in these systems. Okay? And that's because it's known that if you add charges uniformly along the backbone of a polymer, to go from a flexible polymer to a strongly charged polymer, a polyelectrolyte, okay, that the properties of solutions can change dramatically. So the scaling dependence, for example, of the correlation length with polymer concentration, or the relaxation times, or the chain relaxations, or the viscosity. These can be quite different for these two different types of systems. And so we thought, and it's because the polyelectrolytes exhibit this more extended conformation due to the electrostatic repulsions along the backbone. So we thought, can we vary the flexibility by looking now at fully charged polymers and looking at solutions of polyelectrolytes? Okay, so again, we look at new systems. There's a question in the chat. Oh, good. That's taken care of. Okay, so we now look at changing we now look at solutions of sodium polystyrene sulfonate. This is a common model system for charged polymers. Okay, we again pick a high molecular weight so that we're able to access similar radius of gyration in free solution as we do to our particle size, and we look at polystyrene particles of different sizes. Okay. And what I want you to see out of these plots is that if we go to very high salt concentrations, where we largely screen the charges along the backbone, that what we measure for different sizes of particles and different concentrations of polymers is approximately Stokes-Einstein diffusion. Okay, we basically, that this plot is effectively one. But if we go to low salt concentrations, we find something that's rather different from Stokes-Einstein. As we go to smaller and smaller particles, we see that we get this unexpected non-monotonic dependence of the diffusivity that happens at a characteristic polymer concentration of about 10 C star. Okay? And we see this most strongly for this as we go to smaller and smaller particles. Okay. So now, again, another mystery. Why is this non-monotonic? So often when I give a different version of this talk, I'll list all of the different explanations that we thought about in the paper and that our reviewers for the manuscript asked us to think about, but I'll just try to highlight some of the key physics here. And the key physics is that a similar non-monotonic dependence of the diffusivity was predicted by de Gennes in the context of polymer melts. Okay, where the presence of entanglements in those systems led to a stronger coupling back now to the bulk viscosity, or the Stokes-Einstein viscosity of those solutions. Okay. So we have charged polymers; they're not entangled here. But is there something that functions like an entanglement? So to look for this, we now look at the distributions of displacements, which I told you back all the way at the beginning. Okay. Here, I'm showing you the distribution of displacements for 100 nanometer particles for a concentration of 2 C star and one of 20 C star. And what I want you to see is that we can see very different shapes. At 2 C star, that distribution is Gaussian, and at 20 C star, it's very non-Gaussian. So we can characterize then the extent to which that distribution deviates from the Gaussian prediction through the non-Gaussian parameter. And if we compare the normalized diffusivity, okay, which had that wonderful non-monotonic behavior, to the value of the Stokes non-Gaussian parameter, we find that we see sort of similar-ish peaks that happen at a concentration of between 10 and 20 C star. The way that we interpret this is that we think that now there's something that's looking kind of like an entanglement. These systems are not entanglement, but there's some heterogeneity in the local structure that leads to something that effectively functions like an entanglement in these systems. Okay. So of course, charged polymers are now quite different than neutral polymers. I've just given you a rather hand-wavy explanation. So we've also gone to try to do scattering on these systems to understand coupling between their structure and dynamics. Okay. So instead of looking at particle polymer solutions, we just looked at the polymers, the sodium polystyrene sulfonate. We do the same kinds of measurements again. We measure the intensity as a function of wave vector, and we measure an intensity autocorrelation function as a function of wave vector. Okay, cool. So I'm going to show you the data now from those experiments. This is on polystyrene sulfonate with some technical details here. And this is a collaboration with Amanda Marcial and Ryan Pulling-Skutvik. So if we measure the intensity as a function of the scattering wave vector, the upturn at low Q, people largely don't understand, and I'm not going to talk about it. What I will talk about is this peak that appears at Q values of about 0.1 inverse angstroms. Okay, this feature here is called the polyelectrolyte peak, and it measures approximately the correlation length in polyelectrolyte solutions. Okay, and this is seen as the characteristic signature of these polyelectrolyte solutions. Okay. We then do dynamic measurements in the yellow shaded region at this range of Q vectors. We again measure an intensity autocorrelation function, and from that extract out a relaxation rate. So given what we measure, we find that the relaxation rate depends on the polymer concentration. Okay? And we again see that we have some kink in our relaxation rate over this Q range. And that kink now appears approximately at the peak in that structure factor. Okay, so again, we understand this physics. We had to do some machinery to back this up, but this is the same physics we had been looking at before. So if we try to invert our scattering data to get out a static structure factor, this is a technical challenge, but we can model the polyelectrolyte structure with perturbations along it due to the rigidity that's imparted by the charge. And so we back out the structure, which is what I'm showing here, with some fairly significant error bars on it. But what we find is that this peak moves to higher Q or smaller length scales as we go to higher concentrations of solution. And now we do the same thing that we did before. We plot 1 over our subdiffusive parameter on top of that. And again, we see this coupling between structure and dynamics. We find that this now allows us to think about what the effect of the concentration, which is really changing that correlation length, on the structure and dynamics in these systems. Okay? And what's really interesting is we can also ask, how does changing the electrostatics change structure and dynamics? And so the series of data I've showed you on the left was for low salt concentrations. We varied the polymer concentration, but we've also done experiments where we approximately fixed the correlation length. And we varied now both the ionic strength as well as to vary the extent of the electrostatic contributions. And so we're thinking about how to try to now relate these measurements now back to predictions from polyelectrolyte theories that have not yet been tested. It's work in progress. Okay, I don't have much time left, but I want to briefly talk about the effects of particle shape. Okay, and this work was actually very practically motivated. We've had a long time collaboration with my colleague Richard Wilson, who's very interested in lateral flow assays, which after 3 years of pandemic, we're all rather familiar with. Okay? But the way that these work is that reporter particles flow through a porous medium. Okay, I'm showing a confocal micrograph of that process happening in a model system. And Richard's innovation here was to start to think about ways to improve the sensitivity of these assays by playing with the kinds of reporter particles that would be used to generate that visual signal. And the systems that he'd been playing with are particles or viral nanoparticles called bacteriophage. Okay? So these are a particular bacteriophage called M13, which is a filamentous anisotropic virus that's rather flexible. And so we thought, working with him, that these would be very interesting systems now to probe structure and dynamics in our polymer solutions. And this is actually very practically relevant for thinking about assay performance, right? Your snot is a polymer solution. Cool. So we looked at a series of different viruses of different aspect ratios, okay? And hence they have similar persistence lengths. But because they have different lengths and different thicknesses, they have very different aspect ratios. And we went back to looking at our microscopy methods to now look at their transport properties. So we started again with the partially hydrolyzed polyacrylamide, albeit at high salt concentration. Okay. What I'm showing here are distributions of displacements as a function and for now our lowest aspect ratio virus. What I want you to see is that these distributions are slightly non-Gaussian, but the extent to which they become non-Gaussian increases as we go to higher polymer concentration. As we increase the aspect ratio of the viruses, these become increasingly non-Gaussian until when we go to our very highest aspect ratio, they're strikingly non-Gaussian, okay, with extended tails. Why is this interesting? Well, we can make comparisons to spheres, and we can show that we can rule out then structural heterogeneities in the systems. We find only Gaussian distributions for spheres. We also know that these systems aren't entangled. Okay, so there's no hopping or rotation-like mechanism. Instead, what we appear to be probing is a difference in the coupling of these systems hydrodynamically along and normal to the long axis of the virus. We've done experiments where we try to say, is there a characteristic length scale? We see that viruses, the triangle symbols, diffuse very differently than spheres, okay? The more circular or square symbols. And we can approximately collapse our virus data if we think about length scales that are intermediate between the length and width of the virus. And so this is using a length scale that's a sphere of equivalent volume. Okay? Interestingly, if we try to compare to predictions for rigid rods, we can't get collapses. So these systems are very flexible. And so the last little bit I'm going to talk about very quickly is, of course, now what happens when we put our flexible viruses in the charged polymer systems. Okay? And so if we now go back, I'm just going to focus on one particular virus. We now make the same kinds of measurements in solutions with now varying polymer concentration and varying ionic strength to change the effect of those backbone charges. Okay? What I want you to notice is that salt concentration really dramatically modifies the diffusivity of the viruses, okay? And that it's quite different from spheres, which is the open symbols that I've just put on. If we go back and look at distributions of displacements in these systems, we find that we get, again, strikingly non-Gaussian conformations. But it's difficult to make the comparison because by changing the ionic strength in these systems, we're also changing the correlation length. And so we can approximately understand our data by, again, thinking about a normalized scaling parameter and trying to plot things as basically 1 over the correlation length. The hydrodynamic radius is not a function of the salt concentration here. And we see 2 regimes of behavior. At small correlation lengths, which corresponds to high polymer concentrations, okay, we get approximate collapse for different ionic strengths. And if we look at the distribution of displacements, we see that for a fixed correlation length, okay, that all of these distributions have the same shape. Okay. When we go to large correlation lengths, which is the limit of lower concentration, but still above overlap, we start to see differences. And so our hypothesis here is that what we're measuring really are differences that arise now from the electrostatic interactions, which are strongest at lower concentrations, but still semi-diluted. Okay. And so just as a comment, we're also extending these experiments to DNA in collaboration with Ray Roberts and Anderson. But I'm out of time. So let me just give you a quick summary. Nanoparticle dynamics and polymer solutions really provide a rich testbed to probe deviations, to probe a coupling between structure and dynamics of a medium and penetrance. Okay, we find it's not bulk viscoelasticity. Okay, we can see effects that arise from electrostatic interactions. We find that modulating the interactions between particles and matrix can change diffusivity. We find that flexibility and nanoparticle anisotropy alter those coupling mechanisms. And in particular, the anisotropic particles couple differently along a normal to the long axis. But note that all of these systems really tell us that the coupling that we see between structure and dynamics really depends on those system specific properties. So, and so we're always thinking about new ways to change those ratios. So very briefly, here are all the people who contributed again, along with the rest of my group. I'd like to acknowledge funding, you for your attention, and thank you, and I'd be happy to take any questions.
Thanks, JC. If you have questions for JC, please raise your hand and I will call on you. Nancy.
Yeah, really interesting and impressive work. I was curious about the distributions that are non-Gaussian that you observed in a number of systems and whether you've characterized the alpha in those systems as well to see how non-standardly diffusive they are?
Yeah, so let me make a couple, thanks for the question, Nancy. Let me make a couple of quick comments. So the first thing I should have noted is that spheres are Gaussian as far as we can tell in our polymer solutions. So the non-Gaussian behavior appears mostly, spheres and flexible polymers are Gaussian. Let me leave it at that. I showed you one place where we did characterize it. That was for spheres in charged polymers. They're not very non-Gaussian. We have characterized the non-Gaussian parameter here in the virus systems. It's in our paper. I don't actually remember the values off the top of my head. We looked carefully at the value of the non-Gaussian parameter as a function of time. We find that it's approximately constant in these systems. We didn't see strong or reproducible time dependencies. We see potentially a weak difference in that parameter at a concentration of around 10 to 20 C star, which is also what we saw for spheres. This is why we got very interested in trying to probe the structure and dynamics of these charged polymer solutions, but we haven't been able to make a stronger argument than that. But we have looked at this, we just haven't been able to relate this back cleanly to a property of the medium. Does that answer your question?
Yeah, I guess I was just thinking, like, if you have this extra weight in the tails, if that sort of corresponds to like a bit more of an oomph being imparted by the polymers on the particle, for example, in the case of the polyelectrolytes, and whether you might see that as a slightly larger alpha value.
Yeah, it goes, I mean, again, what I've shown you is that it goes up with concentration and it also goes up with aspect ratio. And I'm thinking about the plots as a function of time, and there's not a strong dependence. And so what we've interpreted this as is differences in how the particles here couple hydrodynamically to the medium. But so far we haven't seen predictions that we could test for that idea. But we are looking. So if you or other folks know, we're really interested in thinking about how to make this argument a little bit more quantitative.
Great, thanks so much.
Manasa, will you ask your question?
Yeah. Thank you so much for the wonderful talk. So maybe I missed it. So do you consider or does the charge on the particles itself matter? Because you mentioned about salt concentration, but I remember it was more on what is there on the PSS chains. So I'm curious if the charge on the particles matter in these analyses.
Yeah, so the question is, does charge matter? And the answer is it matters in certain systems and not others, right? And so I'll give you two examples. So here, so in our virus systems and charged polymers, the ionic strength matters because it changes the conformation of the polymers. It doesn't seem to strongly affect, and we're working at a dilute concentration of the particles, so we're not strongly perturbing the interactions as far as we can tell. Now let me give you two other answers. I showed you, which I didn't say this plainly, so that's on me. One case where the charge really seems to matter was in our scattering system, where we had now concentrated nanoparticles. This is in, these are in 2-butanone. And with the fact that we see even at relatively even at sort of not super dilute, but 1 to 5 volume% of our nanoparticles, we see movement in the static structure factor peak. And that tells us that we have a characteristic separation between the particles, and that goes in a sensible way with concentration, it goes down, right? And so the interactions there, when we start to concentrate our particles, now electrostatics in that system become more important. And then let me tell you the final thing, we haven't seen. And so I think here, what we often find is that electrostatics, and I don't want to be too general, play a role once we start to really worry about interaction. So here it's when we increase the particle concentration. The final comment that I'll make, and again, lots and lots of data, sorry. The final comment that I'll make is that, and I love this, right? When we have now, this is now back to viruses and charged polymers, where we're changing the background ionic strength. Okay? When things are concentrated, and when the polymer solution is concentrated, charge doesn't matter. Okay, everything collapses cleanly as a function of correlation length. When systems are concentrated and that they're not dilute, they're above the overlap concentration, but not too concentrated, we start to see differences between particle transport at these different ionic strengths, which again, we've attributed to electrostatics, but we don't have a better way to test that right now. But we think that we're seeing hints of electrostatics now mattering in this regime as well. Does that answer your question?
Yeah.
So again, of course, it depends on system. We see different places, really, and the chemistry, maybe this is my dirty thing of saying this as a physicist, the chemistry really does matter. So we try to think and about how we can control that.
Thank you.
Marco?
Hi, thanks for the talk. For when you move from spheres to your virus works and you start to have elongated particles, there are degrees of freedom associated also with shape, right? So I was wondering whether you have looked at that and there is anything that you can learn from that. So
we can't, so unfortunately, the limitation of what we can do right now, we resolve everything as a very strongly diffusion limited spot. So there is really beautiful work looking at the dynamics of flexible objects from folks like Nick DeFackery, where they were able to resolve, for example, fluctuations, and they could look at coupling of the normal modes of those fluctuations back to the medium. We can't see it, and we also we can't change it directly, at least through the salt concentration. So the reason that we know that, we can measure, for example, a hydrodynamic radius of our objects that's insensitive to salt. What we're trying to do, and it's so far unsuccessful, is that we're working with a synthetic biologist. There's a single point mutation you can make with these viruses that changes the stiffness of the coat proteins. People have done this with FD virus, which is a relative of basically the same thing as M13. And so we've been trying to make stiff viruses, and we don't have good data on that yet. But I think that given the limitations here in the size, without going to much fancier microscopy methods than we can access, but I think it'd be very interesting. The best way that we'll be able to probe the effects of that will be by doing the biological perturbations. But like I said, there's beautiful work, older work from Nick DeFackery and Matteo Pasquale, where they do show differences in that coupling, and they're able to do a normal mode analysis. But that's on carbon nanotubes. Yeah, we'd love to be able to do that. We need to find somebody with a fancier microscope.
Thanks.
I should also note that let me make one more comment. We have preliminary data that I don't trust from older work that suggests that maybe you might be sensitive to those fluctuations and scattering too. And that might be a more appealing way of doing it, but I'm not willing to say anything more than we maybe have measured differences, but not rigorously yet. Does that answer your question? It was an unsatisfactory answer, but it's the honest one.
Quick follow-up on that, JC. Is that something that can be probed in simulation?
Yes.
Yeah.
Well, yes, with a star on it. And so we've talked to my colleague Jeremy about potentially doing that. And so the challenge is now is on how do you measure, how do you make the systems? And so the aspect ratios here, he doesn't think right now that we can get to long enough aspect ratios where we would really start to probe the physics of our systems with sort of accessible simulation boxes, right? Because there's a number of choices we would have to make about the system size and the number of particles, and then the size relative to the polymer chains. I think maybe with slightly faster computers, but we've talked about that. And when we've done the estimates, we can't approach something as extreme as those PF1s. With the kinds of methods that we've been using, which I should note that we've focused a lot on the hydrodynamic methods because we thought we would see very interesting hydrodynamic effects. And of course, it turns out we didn't, but we did implement the fancier methods for that.
Thanks. Silpa, would you like to ask your question?
Yeah. Hi, Jacinta. Thanks for that wonderful talk. So there is an equally competing theory for the dynamics of nanoparticles in semi-dilute solutions, which predicts a stretched exponential dependence of the normalized diffusivity on the ratio of an effective radius to the correlation length. So what was your motive to choose the Rubinstein coupling theory over the other?
So we didn't choose. So we've tested actually fairly systematically. So Silpa, thank you for the question first. So we were model agnostic at the beginning. There's actually a number of different potential predictions here. we tested every model that we found in the literature as of 2015. And we've gone back and occasionally retested models as we build out systems. And so in the essay to our first paper in this space, we actually compare to a variety of other models that also talk about differences in the distribution of the polymers, to the phenomenological models, to models based on obstruction. And we go back and revisit that every once in a while. So we were model agnostic. Like I said, I tried to point out that we've looked at a model that for us seems to best describe that, but we have actually rigorously tested what we've been able to find in the literature. And now I'll note a couple of other things. We've worked a little bit with Sergei Egorov at Virginia, who's implemented mode coupling theories and we find that for some of our, that the mode coupling theory predictions are actually can do a reasonable job as well. So there are details on that comparison in the flexible nano simulation paper. And so, and actually even in Farshad's most recent paper, experimentally, he compared back to that original paper because mode coupling theory provides some additional insights into the hydrodynamics there. So I'm not trying to put a particular model on primacy. I've shown you what appears to best describe our data. I've told you where it doesn't seem to well describe our data. And then just to try to say that we're basically picking out a particular normalized length scale. Does that answer your question? But I can talk more grotesquely about the comparisons that we've made and we've tried to make there.
Okay, yeah, thank you.
Amitesh has a question in the chat. Amitesh, would you like to ask that live? Okay, otherwise I will read it out. He says, thanks for a great talk. How much does the statistics depend on the type of hydrodynamic flows in these experiments? Is it possible to study the effect of turbulence on these statistics?
Amitesh, thanks for the question. So I'm going to say we haven't looked at this all. Everything that I've shown you here is quiescent. What I will point out, and where I think, yeah, I'm going to give sort of an unsatisfactory answer. So the place where I know this has been the effect. So let me think. With the methods that we have, we don't have a good way of probing, for example, the effects of the fluctuations under flow. Where perhaps how flow of a complex fluid perturbs these dynamics is best studied is in flows through porous medium. And there are some nice work both from Sujit Datta at Princeton and Amy Shen at OIST, where they show that roughly that you can potentially start to relate fluctuations that are imposed by the medium structure back to the transport properties of the solutions. That's not quite what you're asking, but that's the best comparison that I know. In some past lives, we had looked a little bit at transport of similar systems through porous media. What we found there was that the structure of the medium played a fairly significant role in the fluctuations. And this is, I think, also what we had one sort of discussion about this. And I think, to be honest, Amy's paper does it better. But I think they're understanding how I'm not going to say this very plainly because I haven't thought about how to explain it. The fluctuations as you flow through a porous medium, you can basically enhance or not enhance them depending on the coupling between the deformation field imposed by the flow, and so you can see, for example, signatures of differences in the statistics when you generate flows that have recirculating regions. And I think that's perhaps where that comes in most cleanly. To my knowledge, there aren't studies that would probe this in the absence of porous media. Does that answer your question? I think, let me try and give a more simple summary, right? I think it's a really interesting question. The only work that I know of that's been done in this space is in porous media, a little bit by us, but much more detailed and more thorough by Sujit Datta and Amy Shen. And there that introduces an additional complication because you get very strong coupling between elastic turbulence and the structure of the medium. Yeah, that was a better answer than what I gave originally.
Yeah. Any other questions from the audience? If not, JC, thank you very much for a very stimulating talk and wonderful.
Great, thanks to
hear about all
everybody.
of this.
Join GSNP.
Yes.